The overall goal of this bioreactor design and experiment is to demonstrate the cultivation of a highly dense mixed photosynthetic community in a novel bioreactor for effective treatment of wastewater.
This method can be used to answer key questions such as how to design and operate algal bioreactors in order to remediate waste streams, as well as how to cultivate algal biomass. The main advantage of this technique is the cultivation of high-density algal biomass in the absence of external separation methods.
We first had the idea for this application when we realized that this high-density bioreactor system was highly successful in growing heterotrophic bacteria from activated sludge in the treatment of organic compounds from wastewater, as well as autotrophic nitrifiers and denitrifiers in the treatment of the nitrogenous wastewater contaminant ammonia. Demonstrating this procedure is Thomas Thompson, a BS/MS student at Drexel University, who's also a co-op and technician in my laboratory.
To begin, place the reactor on a mixing plate and add a stir bar to the reactor. Place the recycle tank beside the stir plate and reactor so that the effluent port of the tank is directed towards the edge of the lab bench. Place the waste container underneath the effluent port of the recycle tank. Then place the feed tank next to the recycle tank. Secure the reactor against tipping with an appropriately sized stand and clamp. Insert neoprene peristaltic pump tubing in the recycle pump and feed pump heads. Install the pump heads onto the pump drives with the screws provided with the pump drives. Next, connect pump A's tubing to the